Good evening to all of you. At the outside, I am very much thankful to Omix Publishing Group for giving me an opportunity in such a grand platform. This is the first time I am delivering an international speech and also a presentation on supramolecular biovectors, a novel carrier for drug delivery and gene therapy. In comparison to conventional polymer, what you are using in for formulations design and drug delivery system, so this supramolecular biovector is mimics to the virus, and it is resembles to the viral structures, having a polysaccharides core materials and binded with a lipid bilayer membranes. So depending upon the structure, flexibility and assemblies, this supramolecular biovector is a very good and novel approach carrier. You can target it into a particular site of the body. Apart from that, supramolecular polymers are made up of repeating unit of monomers linked together by non-covalent bonds. Sometimes the monomers are also linked with hydrogen bonds. As for your requirements, you can break the hydrogen bonds and you can improve the solubility of hydrophobic drugs. As supramolecular polymers have two core, the internal core is hydrophilic and the exterior core is lipophilic. It is highly susceptible to lipid molecules or fat-soluble drugs. In nature also it is available, that is for example polysaccharides, proteins, nucleic acids. This is also best examples of supramolecular biovectors as a carrier for drug deliveries. For example, plastic molecules, if you take a plastic molecules, if you see the molecular structure or nanosite structure of the supramolecular carriers, you'll get this type of structural evidence. Here this is the monomers units joined together by non-covalent bonds as well as the carboxyl groups joined by hydrogen bond. So what for we need supramolecular as a polymer for drug deliveries? Because conventional polymers, they require high pressures as well as temperatures to melt in order to convert the viscosity conformations from high viscous to low viscous. So here, if you use supramolecular as a polymer for drug delivery designs, then it is required very less temperature and as well as pressure. You can design the product or you can develop a new product within a limited period of time. Next, it is ability to self-fill fracture in their structures. So maintain the polymeric properties in solutions also. Apart from that, as it is consists of the different networking structures. So due to the networking structure, you can target it into the particular site wherever you can target because of their mucosal adhesive properties. So you can adhere basically in the nozzles when you are delivering a vaccine drug. What should be the advantages of supramolecular polymer? First is that the novel assemblies over the traditional formulations for the treatment of other diseases like cardiovascular and inflammatory disease should be substantiated by using this particular polymers. Systemic drug delivery can be achieved for controlled drug delivery for predetermined rate release patterns, for example control release or sustained release product. Next, supramolecular biovector is a good potential for mucosal adhesiveness, what I have told just now. That is a very good adhesive properties in the nozzle's mucosa. Next, what is the chemistry of supramolecules? Just like that of lock and key. When you are fixing a receptor in a particular site of the substrate, then it is initiated the pharmacological actions. Similarly, here molecules target or your receptors targeting site to the particular substrate, then only you can design a supramolecules, or you are preferring a cross-linked or linear chain supramolecules depending upon the structures, the functions, the applicabilities, and the targeting also varies. Here the chemistry also related to the, for example, you are checking receptor targeting to a particular receptor in your body, then you are combining with the substrate by the molecular recognitions or by non-covalent linkage. You are getting polymers itself as well as suprapolymers or supramolecules you are getting. Then the different kind of structure of supramolecules, that is linear chain, then it is branched 3D structures, crystal lattice, So many structures you can get by changing the molecular modelings or by change the structure activity of the supramolecules. So this is also another picture of structure of the supramolecules, just like that lock and key. When you are combining a substrate with the receptor, you are getting linear chain supramolecules. If the structure is little bit changed by inserting a ligands or some other part of the substrate, then again you are getting cross-linked supramolecular polymer. Next, what should be the properties of supramolecular polymers? The properties is depending upon the coordinations, pi-phase stackings, number of hydrogen bonds, and the linkage between the monomer units, and what is the length of the monomer units. So depending upon all the factors, all the properties, you can design a formulation. Next, these are the outward shape of the supramolecular or crystal lattice structure, or different form of the structure of supramolecular polymers. So like that of crystals, you are getting different type of crystal structures according to the crystal habit, then crystal angles, then crystal lengths or lattice lengths. Similarly, the outward appearance also varies depending upon the crystals or monomer unit length, or the number of hydrogen bonds, number of carboxylic groups attached to the particular polymer groups. Next, you will see the self-organized supramolecular polymers, how it is changed, the structure, how it is from smaller size to bigger size. So this is one of the transformations, reversibly transformation of the three different keynotes. Here one structure combines with another's, like that of substrate with receptors, ligands. Finally you are getting a supramolecular biovectors, and you are using as a carrier, nano carrier for drug targeting or gene therapy. This is also another structures. Then what should the mechanisms of supramolecular polymerizations techniques? Three mechanisms is involved. One is isodesmics, second is nuclear elongations, third one is ring chains modifications. In the first case, what will happen? The number of chain monomer unit is gradually increases by linking with the hydrogen bonds or non-covalent, reversibly non-covalent bonds with one monomer unit, the another monomer units, and they are gradually increase the size of the polymers. That is one of the mechanisms. Second is that nuclear elongations. They're just increasing the length of the polymers from one to another. Just monomer is gradually joined by their bonds with hydrogens as well as reversibly non-covalent bonds. Increasing not only the size, it also shows a particular structure. So what is the fabrications or design of the supramolecular hydrogels? The polyethylene glycols, fluorescence isothionates labels dextrans, ethylene caprolactans, stannous to ethyl hexanoate, dodecanoate, toluene, tetrahydrofuran, dichloromethane, N-N-dihexyl carbamide, 4-N-N-dimethyl aminopyridines. So these are the basic requirements. Whenever you are designing a supramolecular polymers, you have to use these materials. Thereafter, you prepare a suitable concentration of the polymer. Then you go for incubations along with continuous stirring. You will get a supramolecules. But you have to characterize whether it is within the range of 10 to 30 nanometer or not. So these are the basic compositions, gel compositions to prepare a supramolecular hydrogels. So here the composition is, the mixture was vigorously stirred followed by sonications for 2 minutes, then incubated in a water bath at 37-degree temperature for few minutes. Though the self-assembly of the alpha cyclodextrin and methyl polyethylene glycol and polycaprolactans and trichropolymer inclusions complex, then the trichropolymer complex is nothing but that is the hydrogels, supramolecular hydrogels. So these are the different compositions of different viscosity grades by taking trichropolymers. So this is the chemistry of the synthesis of supramolecules, hydrogels. The raw materials you are using for the synthesis of this particular polymers. So similar the structure you see, what you are doing, you are taking the trichropolymers, you are heating up to 60 degrees, gently heating, and along with continuous stirring with mechanical stirrers, and the structure is little bit changed. So different color you are getting. So the yellow color is the water, green color is the tetrahydrofurans, then water with or tetrahydrofuran is the different colors. Along with alpha cyclodextrin complex you are getting, if you see the molecular level of the structure. After preparing the or fabrications of the supramolecular biovector or the polymer, you have to evaluate or characterize. So whether whatever the supramolecular hydrogel you are preferred, what should be the viscosity, whether you have done any rheological study or not, and the viscosity, how it change. Sometimes you are taking low viscous supramolecules, but once you are designing a formulations, the viscosity is immediately increases or increased. What will happen? What should the cause? Everything you have to perform, and you have to find out the requirement of the taste. Is there any polymorphic change? After preparations, crystalline to polymer, that is amorphous. If it is changed to crystalline to amorphous, so the solubility is also changed. Apart from that, you also do certain in vitro studies based on the molecular weight of the supramolecular biomaterials. So these are the molecular weight, increasing the molecular weight, if you find out what is the cumulative drug release, or sorry, cumulative release of the biomaterial from the dextrans copolymer, tricopolymers, by increasing the molecular weight, so there is a retardations in the release profile. Another is intramuscular injections of supramolecular hydrogels. First of all, you are converting high viscosity gels, that is hydrogels, by applying shear, it is converted into low viscous. If you restore up to some period of time, then again it is increase the viscosities. So it is a very beauty of the hydrogels. Because of increase in the viscosities, so there is a very good sustain actions. So the different approaches of supramolecular polymers is that by assemblies through the controlled self-assemblies with photodiodes, using photodiodes, you are changing the, what is the wavelengths from UV to visibles. So once you will change, so whatever the core materials or the API embedded in the hydrogels, it is escape out and it is initiated the pharmacological actions based on their UV range as well as visual range. So this is another delivery approach, that is capsules embedded or the drug embedded inside the hard gelatin capsules. You are cutting the capsules or removing the capsules wherever you are tight along with that supramolecular biovectors, and you target into the particular sites in the different mode or different sites where you are targeting. What is your targeting site? So according to your targeting sites, you are delivering the formulations. Biovectors, it is basically used for sustained release formulations to enhance the viabilities as well as solubility of poorly soluble drugs or API. Supramolecular based drug delivery has still greater potential for many applications, including anti-tumor therapy, gene therapy, radiotherapy, in the delivery of proteins and as vesicles fast in the blood-brain barriers, because protein and peptide are highly susceptible to degradation. If you use supramolecular biovectors, that is polysaccharide cores, along with bilayer lipid membranes, then definitely it will protect the degradations of the protein molecules. Then you can stabilize the protein molecules. So it is an excellent carrier for the delivery or the targeted of protein and peptide drug molecules. So what should be the current and future perspectives of this polymer is that the mechanisms under the cellular uptake of polymers and their intracellular traffickings and fat nets are dead dash. In addition to that, current therapeutics based on polymer assemblies are mainly studied for tumor therapy. The advantages of this novel assembly over the traditional formulations for the treatment of other diseases like cardiovascular, anti-inflammatory disease should be substantiated. Due to reversibility of non-covalent interactions, these supramolecular polymers are dynamic materials capable of rearranging the structural patterns and thus have major advantages over the conventional covalent polymers. So based on their structure flexibilities and the designings and fabrications, it is an excellent polymers for targeting the vaccine drugs or interleukin 2 or deliver the proteins and peptide drugs or unstable molecules. So this is my conclusions, and thank you.
Running short.